Hello everyone, my name is Daehyeong Kim from Seoul National University. Today I will talk about bio-inspired electronic eye and wirelessly integrated wearable and implantable devices. This slide shows my brief biography. I'm working at Seoul National University. I obtained my bachelor's and master's degree in chemical and biological engineering from Seoul National University. And I obtained my PhD degree in material science and engineering from University of Illinois at Urbana-Champaign. I joined Seoul National University in 2011, and I'm now a professor in School of Chemical and Biological Engineering of Seoul National University. Also, I'd like to thank to my funding agency, Institute for Basic Science. Our group has been working on the nanomaterials-based soft bioelectronics, and this slide summarizes our group's previous research. The soft bioelectronics can be largely classified into two research fields, the soft bio-inspired electronics and the soft bio-integrated electronics. And today, among many types of soft bioelectronic devices, I will focus on the recent research outcome of each research field, such as the bio-inspired electronic eye and the bio-integrated wireless drug delivery system. The first part of my presentation will cover the electronic eye, and the second part will cover the wireless DDS. For many kinds of bioelectronic devices, such as autonomous vehicles, drones, and smartphones, a novel wide field of view camera with a miniaturized form factor is very important. But if we take a look at the conventional wide field of view camera, such as DSLR, its size is very large and its weight is very heavy. This is because the optic system is bulky and heavy. And if we take a look at the DSLR, most of the volume inside the camera is filled with the lenses, as we can see in the table on the right side on the slide. However, such a large wide field of view camera module is not compatible with mobile electronics. The large size is not compatible with thin smartphones, and the heavy weight is not appropriate for the flying drones. Therefore, the development of a novel miniaturized, lightweight, and wide field of view camera module is very important. Then let me first explain why the optic system in the camera module should be bulky and heavy. It is because the image sensor array is flat. Typically, image sensors are fabricated on a silicon wafer. Therefore, the image sensor array is rigid and flat. And if we want to make a good focus both at the center and at the edge of the image sensor array without optical aberrations, multiple lenses should be used. In other words, if a single lens is used with the flat image sensor array, a good focus can be made at the center of the image sensor array, but a severe blurring occurs at the edge of the image sensor array. But if we take a look at the human eye, a very good focus can be made both at the center and at the edge of the retina, only with a single lens. This is because the human eye retina has a hemispherical shape rather than a flat shape. Therefore, even with a single lens, a good focus can be made over the entire region of the retina. So we can see clearly an object in the front as well as an object at the side. This means that a novel image sensor array whose shape is curved like the human eye retina is required to miniaturize the optic system and thereby to miniaturize the entire camera module. In the industry, there have been efforts to develop such a curved image sensor array inspired by the human eye. For example, Sony has reported a concept about a curved image sensor array to replace the multi-lens optics system into a single lens system. And other research institutes such as Microsoft Research has also developed the curved image sensor array as shown in the image on the right side of the slide. However, all the curved image sensor arrays developed in the industry have a very slight curvature, which is far from the hemispherical shape of the human eye retina. Although this slightly curved image sensor array is better than the flat image sensor array in terms of reducing the number of lenses and optical aberrations, it cannot achieve the miniaturized form factor and the wide field of view. Therefore, in the academia, an entirely curved image sensor array inspired by the human eye was reported. Professor John Rudder's group has developed a hemispherical image sensor array by using the ultra-thin silicon nanomembrane. Due to its hemispherical shape, the image sensor array was perfectly compatible with a single-lens optical system, and it could make a high-quality imaging with optical aberrations, while it also achieved a miniaturized form factor. However, the human eye has a field of view less than 80 degrees. Therefore, the same group has reported another type bio-inspired image sensor array for the much wider field of view. So, Professor Rogers' group has developed an Arthropod-inspired image sensor array. In this case, the curved image sensor array has a convexly hemispherical shape. And on each image sensor array pixel, a microlens was integrated, and the image sensor array could achieve an extremely large field of view, more than 120 degrees, while its size is much smaller than conventional wide field of view cameras. But such a compound eye type image sensor array cannot achieve a high resolution. For the high resolution imaging, a chambered eye type image sensor array is required, like the human eye. Therefore, to achieve the miniaturized form factor, the wide field of view, and the high resolution at the same time, a novel camera module should be developed. Therefore, we investigated the features of the aquatic vision. The fisheye has a concavely curved hemispherical retina with a single lens, and its basic structure is similar to the structure of the human eye. But its detailed shape is quite different from the human eye. First, its lens has a spherical shape and features a gradually changing parabolic reflective index. These unique characteristics of the fisheye lens allow the fish to have a very large field of view and a very deep depth of field. Its field of view is twice larger than that of the human eye, and its depth of field is very deep, and therefore it can make a good focus for both a close object and a very far object. Furthermore, the lens size is smaller than the human eye lens, so the entire system can be miniaturized a lot. Another interesting feature of the fisheye is its retina with elongated rod cells. Fish lives underwater, and the light intensity underwater is lower than the ground. Therefore, the photoreceptors of the fisheye have a higher sensitivity than the photoreceptors of the human eye. The fisheye has evolved elongated rod cells, and these long rod cells lead to the high light sensitivity. Inspired by such structure and functional features of the fisheye, we have developed a fisheye-inspired electronic eye. An artificial fisheye lens has a core shell structure in which the core and the shell have different reflective index. The core has a higher refractive index than the shell, which mimics the parabolic reflective index profile of the fisheye lens. The integrated spherical lens features a large field of view up to 120 degrees, and its optical aberration is low. The images of the half-ball lenses are shown on the right side. The image sensor array has a mesh structure which can conform to the hemispherical surface. In each crossing point of the mesh array, a pixel that consists of a photodiode and a multiplexing diode is located. All pixels can be multiplexed by using the back-to-back photodiode and multiplexing diode design. Each pixel is connected by the serpentine-shaped interconnection, and this island and serpentine-shaped interconnection design allows the entire array to be stretchable, and therefore the array can be conformally integrated on the hemispherical substrate. For the photodiode, we used an array of silicon-nanorods as shown in the cross-sectional TM image and AFM image on the left side of the slide. The nanorod structure enables a much higher light absorption than the light absorption by a flat silicon photodiode by suppressing the surface reflection and enhancing the light absorption. This surface modification strategy has been used in the solar cell industry, and it also works in the image sensor. Furthermore, the aluminum oxide was additionally coated on the surface of silicon nanorods by using the atomic layer deposition process. The fabrication of the nanorods requires etching the silicon surface, and this etching can generate surface defects that can cause surface recombination of carriers. However, the surface passivation by the aluminum oxide can dramatically suppress such a surface recombination, and as a result, we can reduce the dark current and increase the signal-to-noise ratio of the silicon nanorod photodiode. By integrating the spherical monocentric lens and curved silicon nanorod image sensor array, we could fabricate a miniaturized wide field of view and highlight sensitivity camera module. For the integration, we made a cylindrical housing by using a 3D printer. Due to small optic system with the curved image sensor array, an almost 8 times smaller camera module than the conventional wide field of view camera module could be fabricated. Then we used the integrated camera module for the imaging demonstration. A good focusing could be made both at the center and at the edge of the image sensor array due to the hemispherically curved image sensor array. Also, the wide-angle imaging up to 120 degrees could be successfully made. From dim light to a bright light, the electronic eye camera showed a good imaging performance. We also demonstrated the deep depth of field imaging. With a single image distance, in other words, with a fixed distance between the lens and the image sensor array, we could perform successful imaging for both a close object and also for the far object. That was the end of the bio-inspired electronic part. Now let's shift our gear to the treatment of status of epilepticus, which is an example of bio-integrated electronics. Let me first explain status epilepticus. Status epilepticus, which is also called as SE, is a kind of fatal seizure. SE is a single seizure lasting more than 5 minutes or 2 or more seizures within a 5-minute period. SE is a life-threatening medical emergency, particularly if the appropriate immediate treatment cannot be made. Between 10% and 30% of people who have SE die within 30 days. SE occurs in up to 40 per 100,000 people per year, which makes up about 1% of people who visit the emergency department. Since SE is so fatal, we need a novel device for the immediate treatment of SE. And such a device should be able to monitor the subject EEG continuously and deliver appropriate drugs immediately when SE is detected. To achieve this goal, we can consider either wearable devices or implantable devices. But each type has pros and cons. Wearable devices are non-invasive and easy to control, but they are not appropriate for fast drug delivery due to the skin barrier. Meanwhile, implantable devices can deliver many types of drug rapidly, and therefore they are a very good option for the immediate drug delivery. However, the implantation of the bulky device requires surgeries that accompany a large incision. Therefore, a new healthcare platform is highly needed, which can combine advantages of the wearable device and advantages of the implantable device. Therefore, we developed a biointegrated electronic system that consists of a soft implantable drug delivery device, which is wirelessly integrated with a wearable EEG monitoring device and a watch-type power transferring device. First, the wearable device on the forehead monitors the subject's EEG. If seizure is detected and lasts more than 5 minutes, the wearable EEG sensing device sends a command signal to the watch-type power transmitter wirelessly. Then the watch-type device sends a strong radio frequency signal to the subcutaneously implanted soft drug delivery device. The drug delivery device doesn't contain the battery since we are using the wireless power transmission. Therefore, its size could be miniaturized dramatically, which requires only a small incision for the implantation of the device. Also, the entire device could be made of only soft materials such as rubber and ultra-thin metal films. This slide shows the design of wearable devices. The wearable EEG sensor was fabricated by integrating various chips on a flexible PCB, and it communicates with the wearable watch-type power transmitter through Bluetooth. The watch-type power transmitter was fabricated by integrating chips on a custom-designed PCB and packaged in a 3D printed housing. If the wearable EEG sensor sends an emergency signal to in the case of SE, the watch-type power transmitter received the signal, and then it sends a strong RF signal to the soft implantable drug delivery device. But if the wireless coupling between the wearable power transmitter and the implanted drug delivery device is not good, the power transmission efficiency may not reach the maximum value. Therefore, to make sure a good coupling, two LEDs were used to indicate the good alignment between the wearable power transmitter and the implanted device. If good coupling is made, both red LED and green LED are turned on, and under this condition, the maximum power can be induced in the implanted device for the drug delivery. If the drug release is successfully made, only the red LED is turned on. When a strong RF signal is transmitted to the drug delivery device, the power is delivered to the platinum electrodes inside the drug chamber. Then the water contained in the drug chamber is dissociated into oxygen and hydrogen on the platinum electrodes, and therefore the internal pressure of the drug chamber increases due to the generated gas bubbles. And again, due to this increased internal pressure, the drug outlet is ruptured and opened, and the drug release is started. After approximately 3 minutes, all drugs contained in the drug chamber can be released, and the drug released from the implanted device in the subcutaneous region will be delivered to the bloodstream immediately, and as a result, SE can be suppressed. We confirmed the safety of the drug delivery device through a series of tests. First, the RF microwave safety was confirmed by measuring the specific absorption rate, which was below the federal regulation. Also, the mechanical stability of the device was confirmed through bending and pressing experiments. Even under severe repetitive bending and pressing with heavy weights, we could not see any mechanical fractures or failures of the drug delivery device as shown in this slide. We tested the system by using a mouse model. SE was induced in 12 mice in total, 6 mice for the negative control group and 6 mice for the experimental group. In the experimental group, the system monitored EEG, detected the seizure, and diagnosed the condition as SE when the seizure lasts more than 5 minutes. And finally, the subcutaneous drug release was made by transferring a strong RF signal wirelessly. In the control group, no treatment was made. As a result, all mice in the control group died in 24 hours, but all mice in the experimental group survived from SE even after 72 hours. We also fabricated a scaled-up version of the implantable drug delivery device. For human application, we can either increase the concentration of the drug or increase the amount of drug. In this slide, a scaled-up version of the device that can contain a larger amount of the drug is explained. To demonstrate the potential for the human application, we tested the drug release from this large-scale device by using the pre-recorded seizure signal obtained from a human subject. A large-size device was designed to contain 1 milliliter of the drug, which corresponds to the clinical standard of the first-line treatment for patients with status epilepticus. The device was tested by using an ex vivo model, and the wireless actuation for the drug release was successfully made, and the drug release in ex vivo model was successfully demonstrated. This movie summarizes the overall scenario of the occurrence of SE and the immediate treatment by the wearable and implantable devices. First, the drug delivery device is implanted in the subcutaneous region of the patient, and the wearable device monitors EEG. When SE occurs, the watch-type power transmitter sends a strong RF signal to the implanted drug delivery device to release drugs to the bloodstream immediately. Then the seizure can be suppressed and the patient can receive an appropriate follow-up care. All such device functions were demonstrated through the animal experiment. The control program was custom-made by using LabVIEW software, and the program controls the entire system. When SE is detected, a wireless power transfer is made to release the drug subcutaneously. And finally, the seizure in the mouse model could be suppressed and SE could be stopped. In conclusion, our group has been working on nanomaterials-based soft bioelectronics. It can be either soft bio-inspired electronics or soft bio-integrated electronics. We typically first design nanomaterials. Then we choose appropriate assembly method for the synthesized nanomaterials and fabricate each device component, including sensors, actuators, power supply modules, wireless modules, and other electronics. Finally, we integrate all such device components into a system whose structure is optimized for the specific application. This is the end of my presentation. And if you are interested in more details, please visit our website to check each publication. Thank you very much for your time and attention. Thank you.